We have a lot to cover for this next exam. Chapter 4, we're finally moving into the cellular level here, but the cellular level is going to include the organelle level. I'll talk a little bit about prokaryotes, but I'll focus mainly on eukaryotes. Okay, but just some general information is what we start off with. So I edited some of the PowerPoints if you happen to look at them earlier, a little bit of change, but in your notes, I talk about cytology, cyto meaning cell. So you'll see this term when we talk about cytoplasm, cytosol, things of that nature. Logi just means study of, so it's talking about the study of cells. What all these cells have in common, okay, is that they're mammalian cells, or to simplify it, they're cells found in the human body, all of these. Okay, but you can see they all don't have the same shape, but they're considered cells, so there must be some type of homology or similarities between them. So in cytology, we study the cellular structure, and then based on that, when we look at the cellular structure, then we move into a function. So what we'll focus on heavily in this class is cellular structure, specifically organelles, and then this is an organelle, but we often refer to it as just a cell or plasma membrane, okay, unto itself, but it does function as an organelle. When we go over these organelles, including the cell membrane, this is where you're going to need to know your organic compounds again, especially lipids and proteins. So make sure you write down to review lipids and proteins from chapter 3. Now, as I said, though, we don't just look at cellular structure during this cytology, but we also look at function. So what I'm showing you here is the epidermis. You know the epidermis simply as your skin. Okay, your skin's often referred to as an organ because it's made up of two types of tissues. Cutaneous membrane. The epidermis is made up of epithelial tissue. The dermis is made up of connective tissue. The two together form your skin, or what we call the cutaneous membrane in anatomy and physiology. And researchers place an emphasis on the epidermis and the type of cells that form it, and the shape these cells take on as they move from deep towards the surface of your skin, which would be up here. And that's when as they follow the change, they not only see the cell structure in the beginning deep, but the cells will change their behavior and then change their shape. And researchers notice, well, those cells, these epithelial cells at the epidermis, their primary function is to function as a physical barrier, protecting other cells of the body. Likewise, we place an emphasis on diversity when we study cells. We not only study individual cell structure and function, okay, we compare different cells to each other. And what they found, say in the case of our body, okay, is that cells have different shapes, and those shapes are tied into their functions. So there's diversity, but similarities. Similarities are homology between the two. Is this little pill right here and this right here? That's both the nucleus. Both of them have a membrane, but the membrane on this cell is stretched out. The membrane on this cell is described as being kind of in a branch shape. These cells here, well, they're cardiac muscle cells. They have a striated look on them. These cells here, okay, have one end that's very, very long. Could stretch out from your spinal cord all the way to the tips of your finger, depending on what type of neuron it is. And then function-wise, these guys are communicators, and they communicate to regulate body functions. These guys, one job, okay, to make sure the heart functions as a pump to pump blood through our circulatory system or through our blood vessels so that our overall cardiovascular system can function as a transportation system. Now, cells, okay, described as not easily or not, you can't see them with the naked eye. So we had the development or invention of the microscope. And the microscope, the most popular one, is just called a light microscope. This is similar to the one we would have used in the lab. Okay, microscopes allow you to perform two functions. Okay, well, you can observe specimens, or in this case, cells that can't be seen with the naked eye or seen easily. So the first function of the microscope is magnification. Just simply increase the size of the specimen, enlarge it. But if you wear glasses or contacts, you know when you're not wearing. Oh boy. Can y'all hear me at all still? Okay, my end's cutting out. Okay, can y'all hear me now? Okay, I lost y'all for a second. I'm sorry. It looks like we're going to have a rough morning today. God, I'm going to have to redo this, and I'm going to hate that. Okay, let's get going here. Okay, so for the microscope, the first function is magnification. Increase the size of the specimen. But as I said, if you wear glasses or contacts, you know if you're not wearing them, you might be able to see an object, but you can't make out any details. So the second function of the microscope is resolution. The technical definition for resolution is the ability to distinguish two entities as separate objects. Okay, simpler way to describe it is to be able to focus or see clearly the details in your specimen. Okay, and in the lab, what we would go over, now we'll save this for later, but in the lab, I'll talk about, let's see, my little hand's not working here. In the lab, okay, I'll go over on the lab overview the parts of the scope that are responsible for these two functions. So down here it says microscopes allow for viewing cells or specimens not seen but with the naked eye. This is giving you an idea of the range, okay, of the size of cells. Animal cells right up here in the micrometer range. Bacteria down here, much, much smaller. If you look down here, just in case you're interested, here's viruses. Okay, so you might hear some talk about you got to wear a mask right now due to the pandemic. But what a lot of people aren't comprehending is that when you wear the mask, you're protecting the person next to you. But if they're not wearing one, they're not protecting you because viruses are so much smaller. We'll talk about these guys later in the semester. But this gives you an overall idea of cell size. Now, there are other types of scopes, and where I worked previously, my students would work with these. We call them phase contrast microscopes. Now, they use a white light source like a light microscope. That's what we call them, a light microscope. And they magnify, and they also perform resolution. But we call them phase contrast because it allowed us to look at living cells. These are actually living cells here. Here's the cell membrane. This little isolate-looking thing here is the nucleus. So the nucleus is not always perfectly round like they show you in textbooks. This right here is the container or substrate that these cells have actually spread out, flattened out, and grabbed onto. So the cellular level is where life begins. So cells are often referred to as the basic units of life because the simplest known living organism or the simplest known form of life is at the cellular level. If we take the organelles that are assembled together to create this form of life called a cell, those individual organelles by themselves, such as the nucleus, the mitochondria, the membrane, they won't do anything. They'll just degrade. That's it. But this phase contrast, it allows you to look at living cells because most often our cells are transparent. Again, had we met in person, we probably would have did a cheek cell stain, and that would have made it stand out more to you that our cells are transparent. They're not like you see with prepared slides where they stain them. And a lot of students will think that the nucleus here is the cell, not realizing, no, it also consists of this outer area, but it's just transparent. Phase contrast microscopes help you get around that. Then we have this little guy called the euglena. It's a unicellular eukaryotic protist under the kingdom Protista. It has organelles called chloroplasts, which contain chlorophyll. So this little guy we would have looked at, and you would have been able to see just a green little thing. He'll change shape. He'll look pill shape, and then he'll round up. He has a little red eye spot made of sensory receptors that help him detect light because the chloroplasts have chlorophyll. Okay, they're able to harness light energy. Here they're showing you the flagellum here, but we usually can't see that in class, but we can see these little guys move around. So this is how it would look under a light microscope, and we don't have to stain these guys. You look at them live, and so some organisms have some color to them, and they're easy to see and track down, as long as they don't move too fast. But then we have these scopes, EMs, or electron microscopes. Light microscopes rely on a white light source to help contribute to you being able to see a specimen. Okay, electron microscopes use an electron beam to help you see the specimen. They're very, very expensive pieces of equipment. It's extremely rare that anybody ever sees one or has access to one. I've never seen one. Okay, the universities I've worked at, you've always had to have special permission to see them. And then to look at the specimens, it's not as simple as using a light microscope, including a phase contrast. You have to prepare the specimens differently. But the detail that these electron microscopes give you is unbelievable. Such as here it says an electron microscope can visualize objects 1,000,000 times smaller than the head of a pin. So they have extremely high magnification strength and therefore resolution. Here's that same little guy, that unicellular eukaryotic protist called euglena, but using a type of electron microscope called a scanning electron microscope. There's actually two types, a scanning electron microscope and a transmission, known as EM and TEM. Okay, when you look at these scopes, scanning, well, the scanning EM or the SEM, it gives you a lot of high resolution detail of the surface of, say, an organism such as euglena. Now we can see it's got a little bit of a gray color, but it looks different than the other under the light microscope. We can't see the red eye spot because all we're looking at is the membrane of the organism, but now you can see the membrane is striated. And then that little flagellum that looked like a little hair-like strand it really stands out now. We would never see it this good under a light microscope. That's a scanning. It allows you to see surface details. Transmission, this is the one that they use to help them identify organelles, get an idea of the structure and location of organelles. But this is where you really have to go through the different procedures to prepare your specimens. I've never seen it actually done. This is the same little guy, unicellular eukaryotic protist called euglena, except they've actually sliced the little guy. They've sliced through him. Now you can see what was a membrane of him. It looks striated, it looks serrated now. All these open areas could be anything from water vacuoles to what we call food vacuoles, because these guys live in fresh ponds, so they have water vacuoles, which eject water back out of them to help them maintain water balance. But you can see in more detail. Then from there, the scientists would have to figure out, what am I looking at in terms of openings here or structures like this in terms of organelles? We call this a transmission electron microscope. It gives you the highest magnification and resolution. That's the way I describe it for all scopes, including the SEM versus the TEM. Now, that's what I said. I've never seen the process done, Sabetha. I've never seen it done. I've only heard it described as very tedious. So the schools I've been at, and you know, with the professors and things that do research, they're not all anxious to get in line and work with a transmission electron microscope, much less an SEM. They let somebody else do it. Okay. Okay. What I want to move into now are the two general types of cell. I touched on this briefly in chapter 1 when we went over the cellular level. At the cellular level in chapter 1, I talked about this consists of cells, and cells are described as the basic units of life. Okay, the smallest known or the simplest form of a living organism is a cell. But I also talked to y'all about there are two general cell types, and initially when they were classifying, naming and classifying living organisms, they initially divided them up based on whether they were prokaryotic or eukaryotic. As time passed, they realized, okay, just because these two different species of a prokaryotic organism are prokaryotes doesn't mean they're alike, so we talked about how they're divided into 3 domains. But for now, I want to look at just the two general cell types, okay? The two general cell types, well, living organisms either fall under a type of general cell called a prokaryotic cell, such as bacterium, or living organisms fall under the second type of general cell called eukaryotic cells. Now, instead of saying prokaryotic cells, you'll hear me say prokaryotes, one and the same. Instead of eukaryotic cells, you'll hear me say eukaryotes, one and the same. Well, here what they're doing is they're showing you a comparison between the two. Okay, what jumps out is the difference in size. Okay, prokaryotic organisms are considered very small. If you go back to the slide where I showed you size, they are much smaller, okay, than they are over here versus eukaryotic cells. Okay, but that doesn't mean they're completely different. First of all, they're cells. All living organisms on the face of this earth, okay, either fall into prokaryotic or eukaryotic cells because they're cells. Simplest form of a living organism is a cell. When you look at these two guys here, despite the obvious it stands out as a difference, there's actually a lot of similarities between the two, such as DNA. I don't care if the living organism is a type of prokaryotic living organism or if the living organism is a type of eukaryotic living organism. What you will find in both is DNA. And make sure you know this. The function of DNA in both types of cells is the same. The function of DNA stores genetic information. So as the semester goes, you're going to hear me say all living organisms have their genetic information stored in the form of DNA. Doesn't matter if they're prokaryotic living organisms or eukaryotic. It's been Can any of y'all hear me right now? Okay, I just got booted out. I think everybody else is still here. All right, I just got booted out. It tells me it's still recording. Okay. All right, so everybody, if it seems like I've left y'all and ditched y'all, I haven't. Okay, I just got booted out. That was a first. Usually we all get booted out. All right, let me continue here. So all living organisms have their genetic information stored in the form of DNA. Okay. What else was found is that the other type of nucleic acid is always present, and we'll visit this later in the semester, RNA. The genetic information stored in your DNA, okay, when your cell needs to read that genetic information to make an active protein, okay, it makes a copy of it in the form of RNA. Okay, so you can also add to this list proteins. They do the work in cells, as I said before. Other structures you'll find in common between the two has to do with what we call the cell membrane. They both have, let me just do this. They both have a cell membrane, except when we go over eukaryotic cells, their outer barrier for most eukaryotic living organisms is a cell membrane that we describe often as a phospholipid bilayer. Whereas for prokaryotic living organisms, okay, they have a cell membrane, and it is a phospholipid bilayer. But because prokaryotic living organisms were found to be primarily unicellular living organisms, some of them live in colonies, but I still view them as unicellular living organisms, meaning that any form of life that is prokaryotic is a single-celled living organism. Therefore, it's not enough for them to just have an outer barrier made up of what we call the cell membrane or phospholipid bilayer. They need more protection, okay? For eukaryotes, we're going to simplify it because we're going to focus primarily on animal cells. But there are some exceptions, such as with plants and some types of protists. You'll find an additional layer in addition to the cell membrane, affording protection to the cell itself. Okay, so I gave you some of these similarities. Another similarity is a non-membranous organelle called a ribosome. Okay, you'll find ribosomes in prokaryotic cells. You'll find ribosomes in eukaryotic cells. The function is the same. So when I go over the function of a ribosome in a eukaryotic cell, it's the same function as in a prokaryotic cell. They synthesize proteins. And I already told you, you find proteins in both that do the work. Now, as far as differences go, I'm going to jump over here. As far as differences go, unlike eukaryotic organisms, which not only have non-membranous organelles such as ribosomes, okay, eukaryotic cells have membranous organelles such as a nucleus. For prokaryotic cells, they have non-membranous organelles such as a ribosome, okay? But what they don't have are what we call membranous organelles such as a nucleus. Therefore, for prokaryotic cells, they have DNA just like eukaryotic cells, but unlike eukaryotic cells, the DNA of a prokaryote is not stored in a membranous organelle called a nucleus, but it is concentrated and tightly bundled together, okay, in a region called the nucleoid region. This is where you will find DNA bundled together or concentrated, okay, so it's not flopping around. Now, another difference you will find between prokaryotes and eukaryotic cells are these structures here. This right here, everybody, I tend to say cell membrane to simplify it. My majors, my science majors, I tell them I use cell membrane, but I tell them they need to know plasma because I know the four-year colleges like Texas State and UT, they'll say plasma, they won't say cell. But wherever you see plasma membrane, you're going to hear me say cell membrane. I said this is synonymous. You find it both in prokaryotes and eukaryotes, and it's described as a phospholipid bilayer. In other words, it's primarily made up of a type of lipid called a phospholipid, but you also find proteins, okay, contributing to it. All right, so they're showing it here. It's the deepest layer, this light yellow color. It's facing the inside or facing towards the nucleoid region, okay? But then you find an additional layer. Now, some eukaryotes have this additional layer, such as plants. They have a cell wall just like prokaryotes. But the compounds that form the cell wall of a prokaryote are very different than the compounds that form the cell wall of plant cells. Okay? The cell wall of plant cells is formed by the polysaccharide cellulose. The cell wall of prokaryotes is formed by what I call glycoproteins or proteoglycans. In other words, carbohydrates and proteins. If you were to take a microbiology class, they would say peptidoglycans. And then it could even be more complex for certain prokaryotic organisms. Okay, so the cell wall here for prokaryotic cells, unlike for plant cells, okay, it's not about water balance. The cell wall for prokaryotic cells is actually to help protect the prokaryotic organism. That's its job. If you look up antibiotics such as ampicillin and you look at the mechanism of action of these antibiotics, many antibiotics destroy the cell wall, thus compromising the bacterium that's causing the infection, allowing your body to kill it off. Okay, so this cell wall is wrapped around, and it's in green. It's wrapped completely around the outside of this cell or plasma membrane. Now, for many prokaryotes, they have an additional layer, which is this outer layer you can see here, and you can see it's sitting on top of the green layer. It's also wrapped all the way around. It's called a capsule. It's made up of various polysaccharides, and the function of the capsule, okay, is to help this prokaryotic organism attach itself to surfaces. The surface could be something simply as your desk, or the surface could be tissues inside of your body. Now, some of these prokaryotisms have additional structures extending out. They look like hair-like projections, but they're described as protein fibers, and they stick out of the capsule. They're called pili. Sometimes I call them fimbria, but I use pili. It's much easier. The function of the pili is to further help with the capsule. Okay, it works in conjunction with the capsule because the pili have the same function. They're just to further help the prokaryotic organism attach to a substrate, a growth surface, your desk, your body tissues, and things of that nature. Okay. What I'm going to do now is I'm going to focus on eukaryotes for the rest of this chapter. Okay. So most of the time we'll be focused on eukaryotic organisms, and I'll use us primarily as an example. And I'll tell you, hey, going into the test, just focus on our body, humans, okay, or Homo sapiens. And then here and there, I'll point out, hey, we're talking about a protist or we're talking about a prokaryotic organism. But here, what I'm going to go over first are eukaryotic terms and cellular structures before I go into eukaryotes, okay? Listen carefully. These terms, okay, some of these terms we're going to apply to Chapter 5, Part 1, which is membrane transport. So you have to know these terms. So the environment outside of a cell, okay, is described as extracellular. The abbreviation I use for extracellular is EC. Okay. Now, this extracellular environment, you'll find either other cells that are up against the cell they're showing you here, or this extracellular environment, you'll find a lot of proteins and carbohydrates that'll help the cell anchor itself, kind of like a spider in a spider web. However, what you definitely will find making up part of that extracellular environment, part of it, is what we call extracellular fluid. When we get into membrane transport in Chapter 5, okay, you're going to hear me talk about ECF, extracellular fluid. Okay, this extracellular fluid here, it's a solution. This is my abbreviation for solution. I just put SOL, look, apostrophe in. That's how I abbreviate solution. Okay. Well, what are the two components that make up a solution, everybody? In order to form a solution, what two components do we need to combine? Y'all still there? I'm waiting. Not a rhetorical question. Did I lose all of you? What two components do we need to make a solution? Yeah, we need a solvent. And what else? And solutes. And Can y'all hear me? All right, sorry, I got booted out. I'm trying, everybody. Bear with me here, okay? Let me speed this up. I'm sorry about this. So, this ECF is a solution. It's made up of solids, okay, that are dissolved by the solvent. In this class, the solvent is always water. All right, so this is the extracellular environment. The cell itself is divided into two parts. The plasma membrane, which I said I'm going to use cell membrane, but they're one and the same. And then what is contained within this cell or plasma membrane? We call it the cytoplasm. Everything inside is a cytoplasm or intracellular environment, I-N-T-R-A. Intracellular environment. Extra means outside. Intra, I-N-T-R-A, is a prefix. It means inside or within. So the cytoplasm is the intracellular environment. It's divided up into a solution called the cytosol, except you're going to hear me say intracellular fluid. Okay, so the cytosol is also known as the I-N-T-R-A, cellular fluid. The reason why I'll use ICF is because it's easier for students to remember when you compare rather than saying ECF to cytosol. It works a lot better overall to say ECF compared to ICF. But this is a solution. Just like the ECF, this is a solution. So remember that much at least. This solution, excuse me, called the cytosol or ICF, it surrounds two different types or groups of organelles. Okay, in my day, they just said organelles. But now they've so much research, because of research advancements, they now break organelles up into two groups. There are non-membranous organelles. You will find non-membranous organelles, many in prokaryotes, okay, but that you'll find in eukaryotes, such as such as cilia and ribosomes. However, what you won't find in prokaryotes that you do find in eukaryotes are the other group of organelles called membranous organelles. Now, as I go over these two groups of organelles, I'm going to give you a general description to help you understand why they separate them into these two groups. Okay, so this is how it breaks down. Now, under organelles, for now, you're only responsible for the ones that I've given you, okay, that are there in the table. There'll be a couple of spots where I'll ask, or one spot where I'll ask you to add a little extra for ribosomes. But for the most part, what's there in that table is what I'll test you on. Okay, so we're going to focus now on not just eukaryotic cells, but specifically the kingdom Animalia. We're going to focus on just animal cells, starting with non-membranous organelles. When you look at this figure, what you want to do, if you can print it off or if you can do this, is just circle all organelles I have in the table that qualify as non-membranous organelles. So you can see a few of them I have here that are listed in your table. Cytoskeleton, ribosomes, flagellum, on this figure here. On the next figure, I gave you two figures because each one has something the other doesn't. On this next figure, you can see here they show you ribosomes on this one, and then these right here, it doesn't say it, I forgot to add it in. These right here are microvilli. So if you look at your list, you can see this thing is not cooperating today. Y'all are going to start hearing me curse, and I don't care if it's recorded because this program is driving me nuts. This microvilli here is what I circled. So these are some of your non-membranous organelles. Now, when researchers were studying cells, in my day, they described eukaryotic cells, those that may form animals, as sacks of jelly, and movement inside of the cells are random. Well, my classmates and I were like, that doesn't make any sense. Okay, if cells were sacks of jelly, then we would be one big blob, first of all. Second of all, if movement inside of a cell was random, we wouldn't exist. It's kind of like saying, you know, I'm going to do my assignment, I'm going to complete it, but I'm going to let it happen randomly. There's a good chance it's never going to happen randomly. You're not going to get anything done. So as research advanced, what scientists discovered is, not for prokaryotes, but for eukaryotes. Eukaryotic cells have a skeleton that they call the cytoskeleton. They're showing you here. Now, the cytoskeleton is made up of protein fibers. So listen carefully. I forgot to mention this. For non-membranous organelles, okay, a general describe most non-membranous organelles is they are formed primarily by protein fibers. Okay, so for most non-membranous organelles, the best way to describe them is they are formed primarily by protein fibers. Cytoskeleton is a perfect example. Cytoskeleton was found to be formed by 3 different types of protein fibers. I give you 2. Okay. Some of these protein fibers are called microtubules. Some are called microfilaments. So I'm going to use this slide here. Some are called microfilaments. What you're looking at here on this figure are actual, what we call cell stains, fluorescent cell stains. I'm basically trying to outline the border of one cell here. I have students that would perform a technique called fluorescent microscopy, and they would use these fluorescent tags, molecules that had a fluorescent molecule on one end, and on the other end, the molecule would bind anywhere inside of the cell. You had these protein fibers called microtubules, to give the students an idea of how extensive this part of the cytoskeleton is. And the fluorescent dye would fluoresce red using the microscope. Now in here, it would also show them that these protein fibers do not run through membranous organelles. They go around them or they concentrate on them. I was trying to draw a perfect circle, but the nucleus of the cell is right here. Over here, it gave them also an idea of getting rid of this stuff. It also gave them an idea of how the cell membrane is not like in textbooks, round or column shaped. Okay, it can have an irregular shape to it. So all around here are the edges of the cell membrane. All this is the cytoplasm. This is roughly the nucleus. But then they would use other molecules that had a different fluorescent dye tag to them so they could see the other protein fibers that make up the cytoskeleton. So on this figure, roughly about right here, you have one cell. This little area here is roughly approximately where the nucleus is. This was to give them the idea of the other protein fibers that make up the cytoskeleton, which are microfilaments. Okay, so you can see they're pretty extensive throughout the cell. And here's the nucleus. They don't run through it. Now you got to imagine, okay, that a cell, its cytoskeleton, the eukaryotic cell cytoskeleton, imagine if you were to overlap these colors together to show you, okay, how extensive, okay, the cytoskeleton is inside of the cell. So the obvious function of the cytoskeleton, let me go back, I forgot to put the slide on. The obvious function of the cytoskeleton was structural support, to provide structural support, including contributing to the shape of the cell. Now what I forgot to put the slide in, was they found there was a second function for the cytoskeleton. Those protein fibers called microtubules, they're used like a rail system or a highway to transport substances throughout a cell. So the cytoskeleton alone disproved the 2 points that my professor once said to those of us in lecture. Cells, eukaryotic cells, are not sacks of jelly. They have a skeleton that functions in structural support, even contributes to shape. But that cytoskeleton also disproved the second remark he made, that movement is random in a cell. No, it's not. It's not random. Okay, that part of the cytoskeleton is made up of microtubules. It's used as a highway system. Listen, at the end of your notes, I give you 2 links. The first link has to do with life in a cell. There will be a point you'll see a guy that looks like he's wearing slippers or little booties, and he's walking along this tube-like structure. That tube-like structure are the microtubules of the cytoskeleton. And the little guy walking carrying this big blob above his head, that little guy walking is called a motor protein that walks along the skeleton, okay, transferring substances, okay, on purpose from one point to another point, demonstrating movement is not random at all inside of a cell. The other structures inside of the cell I'm briefly going to mention real quick are microvilli. Microfilaments, the other component that forms part of your cytoskeleton that they show you here, some of these microfilaments will push up on types of cells such as epithelial cells of the small intestines. They'll push up. So now you can see the cell membrane extends out like finger-like projections. Whenever you see this structure with only these protein fibers pushing up on the cell membrane, we call them microvilli. Cells that have microvilli, the most popular location in your body is in your small intestines. Why do these cells in your small intestines have microvilli just on one side? Well, these microvilli, okay, that are formed with the help of these microfilaments, that's to increase the amount of cell membrane or surface area, because food passing through your small intestines will move across these microvilli, and by having more cell membrane present here or greater surface area, it improves the absorption or the amount of nutrients being absorbed. Okay. Now, some cells have longer extensions. Okay. This is looking down on cells lining the respiratory system. These are microvilli. These long hair-like projections here are cilia. The other component of the cytoskeleton called microtubules, they will push up on the cell membrane, creating these even longer-like structures. So what you're looking at here is cell or plasma membrane that's been pushed out, forming these very long hair strand-like or hair strands here, but it's actually cell membrane being pushed out by part of the cytoskeleton, the microtubules. They found that the purpose of these longer, which are much longer, you can see these are much longer than microvilli. The purpose of these is to help move substances across the tissues that these ciliated cells form. So a popular area you find cells that have these very long hair-like extensions called cilia is in the respiratory tract, so that they can constantly move mucus being produced by mucus cells lining the airways, push that mucus out so it'll go down into your digestive system. Now there are cells such as paramecium that have cilia, but in that case the cilia for these guys is to cause the entire organism to move quickly. Then we have a flagellum. You see these frequently in prokaryotes, but since we're talking about eukaryotes, okay, if we're talking animals, the only cell that you'll find a flagellum on is a sperm. That part of the cytoskeleton called microtubules, well, it's those same protein fibers that are arranged differently though than they are forming the cytoskeleton. These microtubules are arranged differently, packed much more tightly, and organized to where they push out on the cell membrane. So all this is part of that cell or plasma membrane. It's continuous. It's not attached in here. There's not a little attachment point. You have the cell membrane here and it extends out because you have a bundle of microtubules pushing out. And what will happen is these microtubules are bundled together in a way that they can cause this entire flagellum, flagellus plural, if you change the A to UM, is singular. These microtubules are bundled together in a manner that they can cause this entire flagellum to move. Therefore, flagellum are associated with the function of causing a cell, an entire cell, to move from point A to point B. Okay, so they act like a motor. Then the last group that we're going to talk about are the ribosomes. Okay. Now, for ribosomes, these are the ones that are going to be the exception, first of all, everybody. Okay, ribosomes are not made up of protein fibers. Okay, they're not made up of protein fibers. Ribosomes are formed by RNA and proteins, but we don't call these proteins protein fibers. Okay, so ribosomes are made up of RNA and protein, but the proteins are not protein fibers. Ribosomes are found in one of two locations. Okay, they're either free in the cytosol or ICF. In that case, we call them free ribosomes. Or they're attached to a membranous organelle called the endoplasmic reticulum. Whenever these ribosomes, which are these little dots, the ribosomes are represented by little dots here in the cytosol or ICF, as well as these little dots here attached to a membranous organelle called the ER. These ribosomes, when they're attached to this membranous organelle called the ER, we refer to these ribosomes as fixed. So based on their location, ribosomes are described as either being free ribosomes or they're fixed. Now, it doesn't matter where they're located and how we refer to them. The function of a ribosome is to synthesize proteins, okay? So then the question is, why two different locations? If their job is to synthesize proteins, why are some of them free out in the cytosol or ICF and others are attached to a membranous organelle called the ER, thus they're fixed? Well, it has to do with where the proteins are going to do their work. Okay, ribosomes synthesize proteins, and if the proteins that the ribosome is synthesizing, if their work is going to be done here in the cytoplasm, then those ribosomes will be free ribosomes. That's one way the cell helps keep the proteins being made organized and in the right location. So any ribosome that's a free ribosome here just floating out in the cytosol that synthesizes a protein, that protein will do its work inside of the cytoplasm, somewhere here in the cytoplasm. But there are those proteins that when they do their work, they're going to do their work in one of three areas, and the conditions are much harsher. Sometimes proteins will do their work in the extracellular environment, okay, where they're released outside, such as insulin. Sometimes a protein does its work, but it's part of the cell or plasma membrane. So part of it is going to be facing the extracellular environment, where the conditions are harsher also. Okay, so they got to be treated a little differently. And then sometimes the proteins that are being made, okay, are going to stay within the cell, but they're going to stay within another organelle called a lysosome, where conditions can be very harsh. So therefore, listen carefully. Fixed ribosomes, okay, these are ribosomes that still synthesize proteins, but they're attached to a membranous organelle called the ER, because the organelle is going to modify these proteins, help prepare them for the harsher conditions that they have to do their work under. You've seen this with people. Some people may work at a nuclear reactor and they have to put on special suits and everything. Other people just work at a retail store. Show up, you're ready to work. Okay? So listen carefully. You have two different groups or categories of organelles, non-membranous versus membranous. I cover ribosomes last under non-membranous to try to emphasize that these two different groups of organelles, sometimes their functions overlap. In other words, sometimes they have to work together. So where I leave off with fixed ribosomes is where I begin with the endoplasmic reticulum for membranous organelles. So listen carefully. Membranous organelles. I want to give you a description of what qualifies an organelle as membranous. Membranous organelles are enclosed structures that have a membrane formed by the same compounds that form the cell membrane. So again, membranous organelles are enclosed structures that have a membrane formed by the same compounds that form the cell membrane. Second point for membranous organelles. Within the membranous organelle, compounds are stored, chemical reactions are occurring. So within the membranous organelles, compounds are stored, chemical reactions are occurring. And we will visit these as we go through the semester, and I'll point them out. Okay. Oh boy, we might lose each other, so let's see how it goes. Okay, so for membranous organelles on this slide, I have smooth endoplasmic reticulum, rough endoplasmic reticulum, Golgi apparatus, lysosome, nucleus, did that wrong. Sorry about that. Boy, I was on a roll there. So all these guys here, oh, this is not going well today. Nucleus and the lysosome. So these are all the membranous organelles, some of them that we're going to talk about here. Okay, so for two of these, if you look, we have the smooth endoplasmic reticulum, which often goes by the abbreviation SER. And we have the rough endoplasmic reticulum, which by now you figure it goes by RER. Both of these fall under membranous organelles described as an endoplasmic reticulum. What they mean by reticulum in particular is they're talking about interconnected, flattened membrane sacs. Okay, so you can see very long, extensive flattened membrane sacs that have ribosomes attached to them. That lets you know you're looking at the rough endoplasmic reticulum. The rough actually came about because scientists said, hey, imagine you're rubbing your finger along the surface. It feels rough because of the fixed ribosomes attached to it. But then there are folds or flattened membrane sacs that are not as long and as extensive as the rough ERs, but they're still flattened membrane sacs, but you never find ribosomes attached. There are no fixed ribosomes attached. That's endoplasmic reticulum that we call smooth. Imagine you rub your fingers along the surface of the membranous organelle and it's smooth. Now that doesn't make the SER any less important than the rough ER. So I'm going to start with the SER briefly. Smooth endoplasmic reticulum's function is to synthesize lipids such as phospholipids. As you'll see when we go over the cell membrane, the compounds that form it, phospholipids are extremely important. Now, depending on the type of cell, the smooth ER takes on additional functions, such as liver cells, called hepatocytes. The smooth ER of liver cells called hepatocytes, they're not only involved in synthesizing lipids such as phospholipids, but they also are responsible, these organelles, for breaking down toxins that you may have ingested in your meal. So the smooth ER is extremely important. But the rough ER in cell and molecular biology classes tends to get more of the attention because it's involved in protein synthesis, and it's where these proteins that are being transported to that you see a lot of information given. Rough endoplasmic reticulum. As the first part denotes the rough, that rough is due to they have fixed ribosomes attached. Therefore, automatically you should know the RER, part of its function is the synthesis of proteins because of the ribosomes. However, the endoplasmic reticulum part here, it performs a second function. Proteins synthesized by ribosomes attached to the rough ER will be modified by the ER part of this rough ER. Okay, one of the modifications, attaching sugars. Another modification, beginning the folding process. Because remember what I said, a protein's function, shape or structure is directly related to its function. So let me say that again without tripping over my tongue. A protein structure or shape is directly related to its function. Well, the ER starts helping out with that part also. So let me jump ahead to some of these right here. So here it's showing you the rough ER and the smooth ER. These little beads here are showing you fixed ribosomes attached to this long flattened membrane sacs called our ER. So protein synthesis is being carried out by the ribosomes, and then the ER modifies these proteins, adding sugars, beginning the folding process. Okay. Any protein synthesized and modified by the rough ER, okay, any protein synthesized and modified by the rough ER will be transported, I'm trying to find the figure I had, will be transported to the Golgi apparatus. Okay, so let me see, I had a better figure here. So I'm going to use this figure here. Okay, here it says rough ER and Golgi apparatus. So any protein synthesized and modified here at the rough ER. Notice they're showing you right here. A piece of the rough ER's membrane starts to form a bubble, and it pinches off. It should look like this, a sphere, but they cut it open right here. But they'll look like this. These little guys here, my gosh, it's just getting worse and worse, everybody. Let's do it this way. These little guys here are called vesicles. Okay, I call vesicles little lipid packages. We'll talk about them more in the next chapter. Okay, you can see here, these little vesicles are actually derived from or coming from the rough ER membrane. A piece of the membrane pinches off, forming this little bubble, which they've cut open. The little beads and things on the inside here are the proteins that were synthesized and modified by the rough ER, and now they're being transported to the next membranous organelle, which also appears like flattened membrane sacs. Okay, but it's separate from the rough ER and the smooth ER. This is called the Golgi apparatus, which is here in a greenish color. Okay. Now, even though the colors are different, that's just to separate which is the rough ER and which is the Golgi. If you look at this little vesicle that pinches off that they've cut open, notice the membrane, or this outer part of the vesicle, when it comes in contact with the membrane of the Golgi apparatus, notice they fuse together, they blend together. Well, I said, membranous organelles are enclosed structures that have membranes formed by the same compounds that form the cell membrane. Well, I've already told you before in chapter 3, and I mentioned at the beginning of this chapter, phospholipids are the primary compounds that form your cell membrane. That means phospholipids are the primary compounds that form the membranes of the rough ER and Golgi apparatus and any other membranous organelle. So this is like putting a drop of vegetable oil into a cup of vegetable oil. Once the vesicle fuses with the Golgi apparatus's membrane, that delivers the proteins from the rough ER to the Golgi apparatus. Now, the Golgi apparatus has two functions. It further modifies proteins. It continues to add sugars, which we'll visit later. But the Golgi apparatus also performs a second function, which is sort the proteins. And what I mean by sort the proteins is the Golgi apparatus will sort the proteins by dividing them up and then sending them in the correct direction to their correct destination. Therefore, some of these will be sorted and become part of another organelle called a lysosome. And we'll go over this in a second. So that's one direction these proteins are sorted. Another direction these proteins are sorted, which is showing you here, is they're inserted into the cell membrane. They become membrane proteins. You're going to come to know them as transmembrane proteins. So I'm not going to try to write it out, but we'll cover this in chapter 5. I'm going to talk to you about proteins that are inserted into the membrane. Okay, these proteins, I'm just going to draw a little kind of thick little line here. We'll talk about proteins that are inserted into the cell membrane, inserted into that phospholipid bilayer to where part of the protein's facing the cytoplasm and part of the protein's facing the extracellular environment. Okay, so you'll come to know these as transmembrane proteins. The third destination is down here. See if I can do this. The third direction that these proteins are sorted in is towards the cell membrane, but they're not inserted into the cell membrane. These proteins undergo a process called exocytosis. Now, to be honest with you, in advanced biology classes, we don't say exocytosis, we say protein secretion. So what you're looking at here is exocytosis or protein secretion. That means proteins made at the rough yard, transported to the Golgi, eventually are released outside of the cell into the extracellular environment. This is the pathway the hormone insulin takes, everybody. It takes this third pathway. Okay. So again, the rough ER synthesize proteins because of their fixed ribosomes, modify those proteins such as add sugars and fold them because of the ER part. Golgi apparatus further modify those proteins by adding more sugars and then sort those proteins by sending them in one of three directions. We'll visit exocytosis also in the next chapter. Next membranous organelle called your lysosome. If you look at your lysosomes, I say the lysosome, it has an acidic pH on the inside. Everybody, pH throughout most of the cell and within the organelles is roughly 7.2, but in a lysosome the pH is 5 because those proteins that are sorted and packaged into this membranous organelle, okay, they are actually called acid hydrolases. These proteins that are found in this membranous organelle called the lysosome, they're like guard dogs, okay? Anything they come in contact with, they're going to tear it up. They're not looking for an ID card or anything. You just got to get up close to them. You may have seen this before. Maybe a friend has a dog that's a very good guard dog, and you go over there every day to hang out, and it doesn't matter. You're still foreign to them, and all they know is that if they ever get a hold of you, they're going to take you apart. Well, these acid hydrolases, their job is to break down and degrade compounds and old organelles. So we'll talk about these later, how with lysosomes, whenever a cell wants to destroy something, get rid of it, okay, or put it like in a garbage disposal, it'll transport it to a lysosome. And once it's inserted into the lysosome, these proteins called acid hydrolases, they will break down and degrade any compound or old organelle that's delivered to them. But they work best at a pH of 5. Thus, they're called acid hydrolases, but they're contained within this membranous organelle because if they get a hold of anything, they'll try to destroy it. So the cell has them compartmentalized. So this is showing you the same thing. Sometimes cells can take in a food particle and then it'll take it over to the lysosome, and these enzymes will break them down. So acid hydrolases are sometimes known as digestive enzymes. Next organelle is the mitochondria. We're going to visit the mitochondria in chapter 6, so make a note of that, okay? We're going to visit the mitochondria in chapter 6. For now, the mitochondria, okay, it's a membranous organelle, but it has two membranes. It has an outer membrane and it has an inner membrane. What they're not showing you here is that these two membranes, can't do this, I'm trying to draw an arrow going both ways. These two membranes are actually separated by a space from each other. Okay. Now the inner membrane, if you cut the inner membrane open, okay, you find an area inside of here called the matrix. So the matrix is the area inside of the inner membrane. Okay, so we'll touch on this briefly when we go in chapter 6 because the mitochondria function-wise, the mitochondria is the main site where most of an energy producing process called cellular respiration occurs. Or simply put, the mitochondria is where the majority of chemical energy called ATP is produced. Thus, the mitochondria sometimes is called a power plant, okay? Because that's where you have most of a molecule called ATP being produced. Now, we'll explain what ATP is and everything as we get there, okay? But it's what we call chemical energy. You know better as potential energy. So this is showing you the function. It has to do with cellular respiration, but we'll go over it in more detail when we get into chapter 6. So for this slide, if you happen to be printing them off or saving them, you can just type in chapter 6. That's where we'll visit this. But you can see ATP being produced here. So if it seems like some organelles I'm skipping over real quick, it's because we're going to go into more detail later. Nucleus here. This is another membranous organelle. It's the most popular membranous organelle. Okay, the nucleus, its membrane is given a special name. The membrane of the nucleus is called the nuclear envelope. Please remember that, because we're going to go into cell division, okay, in chapter 8. We're going to talk about a type of eukaryotic cell division called mitosis and a type of eukaryotic cell division called meiosis, okay? Now, mitosis and meiosis actually are described as nuclear division, okay? So when we go over mitosis and meiosis, which is actually nuclear division, you're going to hear me say nuclear envelope. Some of the slides are going to say nuclear envelope, and you have to remember, as it's showing you here, what is a nuclear envelope? It's the membrane of the nucleus, and it's a double membrane. And this membrane, called the nuclear envelope, has holes in it that they call nuclear pores. What they discovered, okay, is, as I said before, movement inside of a cell is not random. And when you look at the nucleus, the reason for these pores is there are certain compounds and organelles that have to be transported out and into the nucleus. Okay, and if they're not lipid soluble, if they can't easily pass through the membrane part, the nuclear envelope, they'll be transported through these pores. The catch is, and I'm not going to test you on this, but the catch is these pores, okay, are checkpoints. The only compounds and organelles that can move through these pores, they have to have the right ID to go through. So again, movement in a cell is not random. It's restricted, it's in purpose, it's directional. Okay, but there's a lot of regulation and control. Now, inside of the nucleus, you find the compound DNA stored. Of the last terms left, if you look here, it says nucleolus and chromatin. Listen carefully. If you ever see the term chromatin, what they are talking about is loosely packed DNA. And I'll go over this again later when we go over cell division. I'll talk to you about, if you ever see the term chromatin, chromosome, or sister chromatids, automatically tell yourself they're talking about the compound DNA, but because the DNA has a different shape, they give it different names because that shape is related to what stage a cell is in as far as is it resting or dividing. That's where they give the terms. Well, cells that are not dividing, the DNA is loosely packed, taking up the entire nucleus. We call it chromatin. Okay. Now this chromatin, what they found is made up of DNA, is that it's actually heavily packed inside of the nucleus. So when researchers would stain a cell with just a general stain, chemical stain, what they found is that the nucleus of the cell would always stain much darker than anything else. If you recall, I showed you the picture of those cardiac muscle cells, and I told you the nucleus looked like a little islet. Well, the reason why the nucleus would always stain much darker than the rest of the cell is because the general chemical stain they would use to stain the cell was just grabbing onto organic compounds. Well, the DNA here, even though it's described as loosely packed, there's still a whole heck of a lot of it packed in here tightly. In other words, it's taking up the nucleus to the point that sometimes they'll describe it as the nucleus is on the verge of bursting open. So when they use chemical stain, the nucleus will stain very dark, and a lot of students aren't familiar with cell staining. Okay, they'll think that the nucleus is a cell, and it's not. It's just got a lot of DNA, so it stains the darkest. But then they started to notice with some cells, there was a region within the nucleus that would stain even darker than the rest of the nucleus. They came to call this region the nucleolus. So it's not an organelle, even though they're showing you what looks like something solid here, it's not an organelle. The nucleolus is a region within the nucleus, and the reason why this region stains even darker than the rest of the cell is scientists discovered that ribosomes that are free and fixed, they're first assembled here at the nucleolus. Then they're transported out through the nuclear pores to go do their job. So what ended up happening is there's a region within the nucleus where you not only have DNA in the form of chromatin, but you have RNA and proteins being assembled together to form ribosomes. So you have a lot more organic compounds concentrated here than there is already here. So it stains darker. So they came to call it the nucleolus. So later in the semester, we'll talk about the nucleus when we talk about cell division in chapter 8. And then we're going to get into what we call DNA replication and gene expression in chapters 16 and 17. I mean, not 16, 17, sorry, 10 and 11. Okay. And when we get into that, that's when we're going to be revisiting the nucleus. And that's when we're going to be tying it into how it relates to the ribosome. We'll go over a process called gene expression. Okay. So as we go through these future chapters, I'll try to remember, and usually I remember, I'll tell you, hey, go back and refer to this past chapter to review to help refresh your memory, okay? Because remember, this class builds on itself. Okay, the cell membrane, I said, is also known as the plasma membrane, but I use the cell membrane in the test. In your notes, I believe, what I did, if I did it correctly, is I basically gave you an outline. When we look at the cell or plasma membrane, it's made up primarily of 2 of 4 organic compounds: lipids, sorry about that, it was going well till then. That's supposed to say lipids, and proteins. Okay, so remember, in chapter 3, we talked about 4 types of organic compounds: carbohydrates, proteins, nucleic acids, and lipids. Of those 4 organic compounds, I'm going to focus on 2, lipids and proteins. Now, under lipids, how many different types of lipids were there, everybody? In chapter 3, how many different types of lipids? Not even asking you for the names, just asking you 4. Okay, so we had fatty acids, we had triglycerides. What were the last 2 types of lipids? I gave you 2 of the 4. What were the last 2? I said fatty acids, triglycerides. What are the last 2 types? Yeah, you should know phospholipids because I'm giving it to you. It's up here. What's the last group? Steroids, right. Okay. Specifically, not any steroid, cholesterol. So in your notes, hopefully I remembered to give you an outline. If I didn't, I definitely did it in the next chapter we're going to cover in chapter 5. So when we look at the cell or plasma membrane, you got to take from chapter 3 2 of 4 organic compounds, lipids and proteins. And from the lipids, we take 2 of the 4 types of lipids. The main ones are phospholipids. Phospholipids make up the majority of the cell or plasma membrane. That's why we're showing you here. Okay, they form what we call, let me get rid of this. They form what we call a phospholipid bilayer. Form 2 layers of lipids. Outside of the cell, we have an extracellular environment made of proteins, carbohydrates, or other cells, but we will definitely have a solution called ECF. Inside of the cell, we have our cytoplasm, and I said that part of the cytoplasm that is a solution, it's called cytosol, but I said I'm going to use an alternative name. Instead of cytosol, intra, I-N-T-R-A, cellular fluid. So phospholipids are the major compounds that form the vast majority of the cell membrane, but they're not the only ones, but they're the main ones. And they form 2 layers, okay? And when you look at this, these 2 layers that we refer to as a phospholipid bilayer, notice how the layers are arranged. Each one of these, the head with the tails, is a single molecule, okay? A single phospholipid molecule. We have many of them arranged side by side to where the heads are facing the ECF on this side, because the heads contain a polar group that is what? Hydrophilic. The second layer that's formed, it's formed to where it looks like it's upside down compared to this layer. But that's because the heads over here, which have polar groups contributing to their formation, and thus making the heads also hydrophilic, they're facing this side because we have another water-based solution called ICF. But these little things here, what do these little gray things represent that we call tails, everybody? What are these tails? What are they actually that contribute to forming a phospholipid? This is one phospholipid, but what are these here that contribute to making it? Keep in mind, everybody, this class builds on itself, so let me kind of clarify what that means. It doesn't get easier, it gets harder, okay? If you're not staying up with your studies. So what do those two little tail things mean? What do they represent, everybody? Nobody knows? Yeah, fatty acids. There we go. Yeah, I was just hoping for a guess. Thank you, Vivian. Yes, fatty acids. Well, if you recall, the fatty acids are hydrophobic. Okay, it's all right. There's 16 of y'all here, so I was just hoping a few people were here that were listening. Okay, so the tails are referring to the fatty acids. Well, you have to know the structure of a phospholipid. So right now, put down review lipids and proteins, everybody, specifically phospholipids and the steroid cholesterol. Make sure you put that down. Review them. So what you have here are the fatty acid tails that are hydrophobic, but they're part of the phospholipid molecule, which I boxed in. But for each layer, the fatty acid tails that are hydrophobic face each other. This results in the formation of a barrier, a hydrophobic barrier, a water-hating barrier. Why is that important? Because there's a solution outside that the solvent is water. There's a solution inside and the solvent is water. So does this barrier, hydrophobic barrier, that's part of the phospholipid bilayer, does that help protect what's inside the cell from what's outside? Yes or no? Does this hydrophobic barrier here? Yeah, it helps protect what's inside the cell from outside. Listen carefully. Said this class builds on itself, okay? So you want to make sure you review the lipids and proteins. Make sure you study this chapter and put an emphasis on cell membrane, because when we go into chapter 5, that's what we're going to be visiting. But we're going to also visit proteins a whole lot. So this is showing you an actual EM, an electron micrograph of a cell. The two dark lines that you see here that I'm running the fingertip against, those two dark lines represent the hydrophilic heads, made up of glycerol, phosphate, and a polar head group. What seems like nothing, an empty area in between these two lines, those are actually the fatty acids. Remember, phospholipids have two fatty acids contributing to their formation, and those fatty acids are referred to as tails, but they're hydrophobic. Okay. Now, in addition to that, it's not here on this figure, I forgot to put it, but I'll show you in the next chapter. Also contributing to the formation of the cell membrane is another type of lipid. It's the steroid cholesterol, and I did mention this when we went over chapter. I talked about how cholesterol is a precursor used by cells in the human body to synthesize testosterone or estrogen. But I said a second major function, or another major function of cholesterol, is it contributes to the formation of the cell membrane. I'll show you when we get into chapter 5. You'll find molecules of cholesterol, okay, inserted between, or oops, that was wrong. Inserted in between the phospholipids. Okay, they'll be inserted in between them if I don't screw this up, in one layer, or they'll be inserted in between in another layer. So I'll show you in the next class, next chapter. Okay, so they also contribute to the cell membrane structure and function. And then we have these guys. If you notice here, they're showing you these little purple colored guys. They represent proteins. So listen carefully. The other major compound out of the four that contributes to the formation of the cell membrane are your proteins. Okay, there's three types of proteins, and I'll go over it more in the next chapter, but the three types of proteins are known as lipid anchored proteins, which isn't on here, peripheral proteins, which is shown down here, peripheral meaning just next to, but the main proteins that we're going to be interested in as far as function goes, are going to be these proteins. If you recall, I said the Golgi apparatus modifies and sorts proteins, and when it sorts proteins, it sends them in one of three directions. One of those three directions is to insert it into the cell membrane. This is a protein that's been inserted into the cell membrane, and so is this one, but this one doesn't go all the way across. Only these that are inserted and cut all the way through the membrane to where part of them faces, say this is an ICF, and part of them faces, this is an ECF, it tells you here, okay? These are what we call transmembrane protein. Trans means through or across. We are going to focus on these proteins, and I'll show you a figure that shows you transmembrane proteins have different functions. The ones we're going to focus on primarily in part of Chapter 5 are the ones involved in membrane transport. But your proteins are your other second major compound. Okay. When you look at these two key compounds, lipids and proteins, and you look at the types of lipids and the specific types of lipids and specific type of proteins that contribute to the formation of the cell membrane, you can then deduce, or from that emerges, they like to use that now, from that emerges the functions. This right here is that hydrophobic barrier formed by the phospholipid bilayer. Thus, physical protection. Keep what's outside outside and what's inside inside. There is some exception though, what can pass through. We'll talk about that. The functions of the cell membrane has to do with membrane transport for molecules and substances that can't move through this barrier. We'll talk about proteins that don't look like this. They have a different shape, but they'll help ensure substances such as glucose can move across. Other proteins we'll talk about, they function as CAMs, cell adhesion molecules. Whenever a cell is not next to another cell, but it's next to the ECF, that contains not only next to the extracellular environment, that not only contains ECF, but contains proteins and sugar called the matrix. Some of these transmembrane proteins function as CAMs, cell adhesion molecules include. And we'll talk about how others function as receptors so that the cell can pick up on stimuli in the surrounding environment. So they'll have different functions. Okay, they can be deduced based on the compounds, but I'll put more of an emphasis on that when we go into Chapter 5. Okay, I'm going to stop the recording there.